So my name is Mark Daly. I'm with Dovetail Genomics. I've been with Dovetail for, I guess, close to 4 years now. And I'm really super excited to have Harris Lewin in our studio, our virtual studio. Harris Lewin, I've known Harris since I started at Dovetail, and we've worked together on all kinds of different exciting projects. Harris is the professor of evolution and ecology at UC Davis. And he's also the chair of the working group of the Earth BioGenome Project. And the Earth BioGenome Project, as you may or may not know, is a huge undertaking, probably the biggest project, biological project, we're undertaking in history in terms of trying to sequence every living organism. And so, yeah, super excited that Harris is here, and he's going to do a really cool discussion on a Narwhal project that he's been involved with. And it's kind of fitting because yesterday was World Ocean Day. And so it seems kind of fitting that we continue celebrating World Ocean Day today with a discussion of one of the most iconic creatures of the sea, which is the narwhal. And so thank you all for spending an hour with us today. And before I introduce Harris, I just want to go through a couple of housekeeping things and a few slides to introduce you to Dovetail Genomics and de novo assembly. So as I said earlier, there's a poll that's running on your screen. Feel free to participate in that. We're trying to gauge how many folks out there are interested in genome phasing and diploid assembly. Those are two things that I'm sure Harris will address in his talk, or at least mention. But these are things that we're able to do now with the newer technology that I'm going to talk about. Also, the way you put in questions is in the Q&A tab there. You can't, you're all muted, so you have to type your questions into the Q&A. We'll try and get to all the questions by the end of the presentation, but if we do not, I will follow up with all the Q&A typed out, and I'll send that to you by email. So all your Q&A will be addressed at some point. So I think let's start. Good biology begins with a great genome, and since there could be folks on the call here that are new to de novo assembly, I do want to address just some very basic things here. And this, I think, will also help you appreciate Harris's talk more. And I just want to go through a few slides. So proximity ligation, what is it? This is the core technology that kicked off Dovetail in 2013 out of Ed Green's lab at UC Santa Cruz. And the core technology is at Dovetail has always been proximity ligation. And it's kind of the common thread through all of our offerings. So what is proximity ligation? And if we look at this chromosome here, now in fact, this is a little bit misleading because the chromosome is unwinding. But if you can imagine an intact chromosome in the nucleus of the cell, we basically cross-link all the proteins that are on the chromosome together. And then we chop up the chromosome with an endonuclease and ligate everything back together. And you can see that there's parts of the chromosome that are much closer together than other parts, right? So these parts of the chromosome have a much greater chance of interacting by ligation than these 2 points on the chromosome. And so if we imagine this occurring over the entire length of the chromosome, we get this nice curve of proximity ligation interactions from lots and lots of interactions close together, which is up here. And as we get farther and farther apart in three-dimensional space, we get fewer and fewer interactions, but we still get lots of interactions that allow us to essentially scaffold the chromosome from one end to the other. And this technology can also be used to interrogate biological interactions within the chromosome, i.e. 3D epigenetics, and we have various offerings for that as well. So that's a definition of proximity ligation. Just quickly taking you through the history of Dovetail And the reason I do this is we have different acronyms that I want you to understand so that when Harris is talking about them, they'll make more sense. So we started in 2013 by assembling the American alligator genome. And for that, we used our original technology called Chicago, which is an in vitro version of Hi-C. So it occurs in a test tube. And we also used our scaffolding software called HiRise, which Nick Putnam wrote. So Ed Green's lab developed Chicago. Nick Putnam wrote HiRise. And we still use HiRise today, actually. It's our scaffolding software. Then in 2015, we came out with Dovetail Hi-C, which is now an in situ version of Chicago. So now we're dealing with intact chromosomes in the nuclei. 2019, last year, we came out with our newest technology for scaffolding called OmniC, and that's what Harris is going to talk about. OmniC, unlike traditional Hi-C, uses an endonuclease to chop up the chromatin, and so you get far more even coverage, and I'll discuss that in a few minutes. And then this year and looking beyond, we're looking at whole genome phasing and also true diploid assembly. The basic pipeline that we use here for our service projects is using deep pack biosequencing, followed by OmniC for scaffolding, and that builds up your scaffolds up to chromosome scale. Then we do an annotation pipeline, and you end up with a publishable ready assembly. There are 4 main challenges in de novo assembly shown on the left, and these can all impact the efficiency and the performance, but we've managed to develop strategies to get around all 4 of these major constraints. And I think the biggest kind of thing that we can leverage is our experience. And on the right, I'm showing you our tree of life cladogram that has around, I think we're up to around 400 species now. We've assembled at least 2,000 genomes, but these are 400 that our clients have okayed us to publicize. And I welcome you to go to our website and take a look at the cladogram. It's really neat. You can see at the bottom, you know, all the mammals have really big N50s and the plants have lower N50s, but it's really cool to kind of see how some of the toughest organisms we've ever worked with, we can still build really high quality genomes for. So the 2 components of OmniC scaffolding, the wet lab at the top and the dry lab at the bottom here. The wet lab is basically cross-linking the chromosomes, chopping them up with an endonuclease, ligating everything back together, reversing the cross-links, and then sequencing. And we get a library of proximity interactions from very close together on the 3D space to very distant in 3D space. And we use those interactions in our HiRise software that's shown at the bottom here. And HiRise basically takes your draft long read assembly, so in our case PacBio, and the proximity ligation data, and essentially HiRise's job is to start making connections of contigs and scaffolds to build up chromosomes. And you can see on the left, this contact map here on the left, where I show you the scaffolds of the assembly on the diagonal, and each little square is a scaffold. There's literally tens of thousands of these in this assembly on the left. After running it through HiRise, out pop the individual chromosomes on the right, and each of those squares is a separate chromosome. And my final slide is just showing you the power of OmniC compared to restriction enzyme-based Hi-C. And the main take-home message I want to demonstrate here is that the traditional shotgun sequence coverage, which is kind of your ideal, is shown with a dotted line here. And you can see that with single enzyme Hi-C or even multi-enzyme Hi-C, you don't really get a distribution like shotgun coverage. So you do get quite a bit of the genome that's underrepresented and quite a bit of the genome that's overrepresented. And that's because you're looking at restriction enzyme bias, right? So with OmniC, because it uses DNase 1, we get a profile much closer to a shotgun profile, meaning that we get this very even coverage of sequence data as shown on the bottom here. Now, why is that really important? It's because it allows us to capture almost all of the SNPs in the genome. And the SNPs are shown here by red lines. The reason we want to capture as many SNPs as possible is so that we can phase the assembly. And haplotype phasing requires very high call rates for SNPs and very accurate call rate, very accurate calls. And so this is why OmniC is going to enable haplotype phasing. And in fact, even beyond that, it's going to enable true diploid assembly. So with that, I want to get on to the even more exciting discussion here, which is Harris Lewin's talk on the Narwhal and his, literally this has been an odyssey for him and his group. We've followed this odyssey of the Narwhal. It's probably the most, well, I'll let Harris talk about it, but just an incredible odyssey of technology that's been used for this particular genome. And so we're really excited that Harris is here now to take us through this journey. And with that, Harris, I'm going to stop screen sharing and let you take over here.
Okay, greetings to all of you from Davis, California. And thanks, Mark, for the introduction and for the opportunity to talk about some of our recent work on producing and evaluating genome assemblies. We agree with you completely that good biology begins with a great genome. Now, my title today is a bit tongue-in-cheek, but it reflects the question of how to produce the highest accuracy, phased chromosome scale, de novo whole genome sequence at the lowest possible cost. And the reason I'm very interested in this, one of the reasons, is that I chair, as Mark mentioned, the working group of the Earth BioGenome project. And one of the main goals of the Earth BioGenome project, as Mark also mentioned, was to sequence and annotate the genomes of all 1.5 million species of known species of eukaryotes over a 10-year period. So certainly cost is a factor and quality is a factor, because if you want to do this, you don't want to have to do it again. So all of this, the goals of the project, the rationale for the project and the strategies for the project are described in this perspective article. It was published in PNAS a couple years ago in 2018. And if you're interested in the details, you can read all of the details where we were 2 years ago. But the strategy, basically, if it will come up here, okay, there we are. The basic strategy is to do the sequencing in 3 phases, where years 1 through 3 of the project would be producing approximately 9,300 genomes, or 1 representative of each taxonomic family of eukaryotes. And in the out years of the project then would be phase 2 and phase 3, producing sequences for all of the genera and then eventually all of the species. What differentiates, at least in the original strategy, phase 1, 2, and phase 3, is that we were calling for reference quality genomes at the family level. Now, what is a reference quality genome? Well, that depends. That's a mark that is constantly moving because of new developments in technology. And so the community has stepped forward with a set of metrics to describe assemblies that can be used to describe their overall quality. And this is a table that appears in a new paper that was put up on bioRxiv a couple of weeks ago by Arang Ri and the Vertebrate Genomes Project Consortium. And the consortium, of which I am a member, has produced a series of metrics by which genome assembly quality can be judged. Now, of course, the ultimate goal is to produce, a finished genome or a near perfect genome, which would be a gapless genome, all phased and all scaffold, all chromosome length. And while we're not really at the point with most eukaryotic genomes because of their size and complexity, as Mark mentioned, of producing finished genomes, at the time the Vertebrate Genomes Project was launched about 3 years ago, we had agreed on a very ambitious metric at the time, which we now call 6.7 Q40. And what that means, basically, is for continuity, that's the 6. This is a log scale. This would be contigs of at least 1 megabase, scaffolds of at least 10 and 50 of at least 10 megabases, and fewer than 1,000 gaps. The Q40 refers to the base pair quality metric, at least one error in 10,000 or better, and more than 80% of the assembly assigned to chromosomes. And this was a very ambitious objective when we started, but as technology has improved, as you'll see, this has become, and through the great efforts of a large number of people, this has become rather routine to achieve assemblies of this quality. And so how was this done? What is, or I should say what was 2 years ago, the gold standard for creating reference genomes? Well, under the leadership of Eric Jarvis and the Vertebrate Genomes Project, the community agreed on a series of steps combining several methods. Those methods would be long reads by PacBio, linked reads from 10X, optical maps from BioNano, high-C scaffolding, which was done by a number of different providers, including Dovetail, gap filling and polishing with Illumina reads, and then creating draft assemblies, a primary phased assembly, as well as an alternate assembly representing the second haplotype, and finally curation of the primary haplotype manually by the team at the curation team at the Sanger Institute. So this was judged as the absolute best gold standard genomes that could be produced a couple of years ago. And as the work started, we began to uncover many different problems and complexities, but amazingly, with a lot of community support, many genomes have been produced. The first 16 species representing at least one of all of the vertebrate classes is contained in this nice paper, which is on bioRxiv, as I mentioned, by Henri, and that has been now submitted for publication. And as you can see, the project very easily met the metrics that we set for the contig and 50 of 1 megabase, scaffold and 50 of at least 10 megabases, as well as the other measures of accuracy and completeness. Not quite as good as we would have liked for all the assemblies, but nearly approaching and nearly meeting the QV40 assembly, QV40 quality metric as well. So I can tell you that, now the VGP has produced over 100 assemblies using this pipeline. They will continue to produce at least one representative for each of the vertebrate orders, approximately 260 reference genomes using this pipeline. And having worked in the field since the first genomes were produced now approximately 20 years ago, I can tell you that these are really the best genomes that I have ever worked with. But I can also tell you that these genomes are not perfect. So how do we know that they're not perfect? And in the lab, Joanna Damas, who's a postdoc, who's well known to many of you, developed a series of scripts to evaluate the accuracy of the assemblies and to look at what was going on at each step of the assembly process. So there are three main processes that occur at each assembly step. So if we think as an input assembly, say this was PacBio assembly, and then we add, say, the next step, the 10x reads, what happens when you add 10x? Well, you can have three possible outcomes. You can have end-to-end joins, excuse me, end-to-end joins of scaffolds. You can have scaffolds that go in as one piece that are broken by the assembly process into two or possibly more scaffolds. And you can have inconsistencies produced so that a scaffold is broken, but it is connected in a different arrangement. So I hope that's clear to everybody, because that is one of the fundamental ways in which we can assess what is happening at each of these steps in the assembly process. Now, these are the data for all of the assemblies in Arang's and the VGP paper, evaluated using Joanna's algorithms, Joanna's scripts. And there are just three steps here shown. The first is what happens when you add 10x to PacBio. The second is what happens when you add BioNano to the PacBio plus 10x. And the third is what happens when you add Hi-C to the PacBio plus 10x plus BioNano. Okay, and it's important to know that as you go across here, each of these end-to-end joins, inconsistencies, and free end breaks are unique. These are not overlapping, okay, because each step in the assembly process was evaluated independently. And what you can see is what you probably would have expected to see as you add a set of linked reads. You're increasing the number of joins, end-to-end joins, and you can see these range in the tens to hundreds for each of the individual assemblies. By adding 10x, you're also introducing some inconsistencies. So some scaffolds are joined in new ways, but adding 10x did not change, did not produce any broken scaffolds with free ends. They would join either end-to-end or they were inconsistent joins. As you move across the table, you see adding BioNano, again, a series of joins, sometimes many joins, for example, in the two-line Sicilian, and now occasionally in some of these assemblies starting to introduce free end breaks. Something that was very interesting to us as you move toward the longer range scaffolding with Hi-C, you can see again additional joins, but now you're seeing more inconsistencies and many more free end breaks. And so the question here is, why is this happening? Is this because each of these steps, it's a very complicated pipeline, and along each step of the assembly pipeline are errors introduced into the assembly that later need to be corrected by the addition of Hi-C. And what I don't show you here is a column which is the manual curation step, which involved correction of tens and even hundreds of scaffolds looked at manually, which were flagged in this particular process. And so, again, there are problems that are introduced at each step that need to be fixed at later steps, and hundreds of scaffolds need to be fixed or manually adjusted. So with all these steps, we began to ask ourselves, and many of the steps introducing errors at each step, is there a way to simplify this process that will allow us to maintain or even improve accuracy and reduce costs? And I just want to say again, these are still the best assemblies that we've ever had to work with, but like any assembly, they're not perfect. And we want to know where those errors are, if they're systematic, and how they might be corrected and improved by the different steps in the assembly process. Okay, so we again want to see, because technology is rapidly evolving, the assembly of algorithms are evolving, and that has created new opportunities to eliminate different steps in the assembly pipeline, and then to evaluate how well we are doing as compared to the more complicated and multi-step process. So as Mark mentioned, I am going to talk to you about the work that we have done over the period of the last few years. I've been working with Dovetail really since the company has founded. It's been really great to watch the growth of the company and the fantastic technology that Dovetail has developed, that has really assisted the community, and particularly my own interest in studying chromosome evolution. And so the work I'm about to describe is done primarily, again, by Joanna Damas in my group, but it's also a larger collaboration involving Martin Narwhal, who is the head of all things Narwhal. Martin's at the Harvard School of Dental Medicine. The Narwhal, as excuse me, Mark mentioned, is sometimes known as the unicorn of the sea because of this large tusk, which is in fact a tooth. Many fascinating aspects of the biology of this animal, and Martin Narwhal has been the absolute long-time leader in the field of studying narwhal biology and narwhal genetics. We've been very privileged to work with Martin over the course of the last few years on this particular genome. Also, Kirsten Howe, the Sanger curation team, Jonas Korlach at PacBio, and the entire Dovetail Genomics assembly team. So what have we done with the Narwhal genome? Well, first, we wanted to see what an assembly that used linked reads. And as you recall, the first technology for Dovetail was the Chicago library. So we had 10x plus Chicago. Then we added 10x Chicago and Dovetail. And then we could add some gap filling with PacBio and a manual curation step. And this was the combination that produced what we call the first curated assembly for the Narwhal genome. In the past year, we've added long reads, PacBio, and we've also added the Dovetail OmniC. And we've done all permutations of assemblies and sub-assemblies so that we can understand what happens at each step. Okay, so thanks to the great assembly team at Dovetail, we were able to evaluate 8 different permutations of assemblies produced by these different methods. Now, I understand that this is a very complex slide with a lot of data, so the stuff that's highlighted in red is really what I'd like to call your attention to. So first things that you will notice is that as we have expected, as you add in the scaffolding technologies, you increase the N50 scaffold size, which begins in the 10x assembly at about 22 megabases, clearly exceeding the VGP EBP standard of 1 megabase, but not exceeding the contig N50. Here, the N50 contig size not shown is about 250 kilobases. The highest contiguity assemblies are produced by the combination of 10x plus Chicago plus high C. That's what we had at that time, plus the gap filling with PacBio and curation. So this is the assembly that we're going to use for all of the comparisons of the different assemblies and sub-assemblies, and basically because it came first before the others. And as you can see, still a relatively large number of scaffolds, nearly 7,000 scaffolds, but very high contiguity, nearly 108 megabase and 50, with an L90, which is very close to the number of, trying to get this out of your way, very close to the number of autosomes in the species. So the Narwhal has 21 autosomes plus a pair of sex chromosomes. And as you can see, this L90 is very close to the number of autosomes. The characteristic feature, I would say, of these assemblies, because they are not longer read assemblies, is the high numbers of gaps and the fraction of the genome that are in ends that are placed in these gaps by the assembly algorithm, particularly the 10x scaffolding method. So about 0.5%, or about 11.5 megabases, are ends in this genome, and these are things you'd like to get rid of if you can. So again, I'm going to show you about 20 different assemblies. They're out of the 28, and I'll just go very quickly to say that PacBio plus Chicago plus IC, or the PacBio, and these are phased using Falcon phase, or PacBio plus OmniC, produce highly contiguous genomes with very similar L90s and N50s, but a far greater reduction in the total number of scaffolds and the number of gaps, and basically 0% ends. If we add 10x to this, we really don't achieve much improvement. We reduce slightly the number of scaffolds, but the N50 and the L90s remain, and the number of gaps remains basically the same. So what have we learned from all this? Well, the assembly sizes overall for 10x and PacBio assemblies are very similar, but the major differences, of course, is the 10x based assemblies are much more fragmented and have a higher percentage of assembly with gaps and ends. No surprise to most of the genome aficionados that are probably listening to this call now. In combination with either 10x or PacBio, it's very clear that Hi-C or OmniC yield the genomes with the highest continuity and L90s that are similar to the haploid number of chromosomes of the species. Adding 10x to PacBio based assemblies does not appreciably change the continuity of the assemblies. But importantly, going into this, we wanted to know if the VGP pipeline could be simplified so that we could meet the metrics. And the answer to that, at least at the, just looking at the N50s, if you have N50 NV, is the answer to that is definitely yes. You could certainly eliminate the 10x. Optical maps were not used in our comparison, although we are adding the optical maps now. But you could eliminate at least the 10x and the polishing steps and still come up with metrics that are very similar to what you would get if you have all the steps in the pipeline. What we don't know, though, is how the removal of these different data types affects the overall quality measures, and that, of course, needs to be determined. So we have done that, and the way we've done that is we have looked at BUSCO. So we evaluate gene completeness by doing a BUSCO assembly analysis of all of these assemblies. And as you can see, there's not much of a difference at all in terms of complete single copy genes, about 90%, 6% for all of these assemblies. So the 10x in this case is really actually unique, I think, maybe, in producing very high quality in terms of its completeness, genome completeness. But the PacBio assemblies, if you look at the numbers, have slightly higher single copy and also lower numbers of missing or duplicated BUSCO genes in the set. Now this can be evaluated, and that's for every one of these sub-assemblies that were evaluated, all 28 or so. That could be evaluated further, and we can see, again, this 96% number, tremendous agreement if we compare the two most contiguous assemblies with a 10x base or a Falcon phased assembly. And we have, 3,000, basically, 3,945 where there's complete agreement between these two assembly methods, vast majority. And as I mentioned, in some cases, the PacBio seemed to be better in that it produced more single copy genes and less fragmented missing genes. But the converse was also true. In some cases, the 10x based assembly produced more complete gene models than the PacBio assembly. So we wanted to look into that a little further. And what you see here is the way we evaluate assemblies using the evolution highway tool. These are sort of the linear arrangement. What you have is, as the reference here, is the curated assembly, the 10x based assembly, and we overlay the Falcon contigs, which are shown in these numbers, on top of the 10x based assembly. And anytime you see a number here, that means in the Falcon, there's a single, if there's one number, there's a single Falcon phase contig that covers that entire chromosome that was produced by an X-based assembly. If you see a little gap, there's a gap there. And that means that piece was actually missing in the 10X-based assembly. And so we asked where those genes are that are missing. And in 10 cases, we could find genes in the gaps. Most of the cases, we found genes that were in the regions of alignments between these two assemblies, which indicates that you really need to look very carefully at missing BUSCOs because there could be some issues relating to how BUSCO is calling either the presence or absence of the gene. And this, of course, is extremely important if you're interested in gene gain and gene loss in evolution. Okay, another very complicated data slide. And this is looking again at the sub-assemblies. And what we're really trying to show here is that there's a huge difference between the PacBio long-read contigs and the scaffolds produced in the NX. So even though the genome statistics look very similar in terms of N50, the fine structures of the order and orientation of the scaffolds within the assembly differ quite a bit between the PacBio assemblies and the 10X-based assemblies. And that could be in the order of magnitude of hundreds of scaffolds that have inconsistent joins when they're compared to this curated 10X assembly. So that was a very important result. Many additional end-to-end joins, quite a number of broken scaffolds, and importantly, it's not just the number of scaffolds are broken, but it's how many times each scaffold is broken. So, for example, in this case, there are 414 breaks in 28 scaffolds. And I guess the important thing I want you to take home is looking at the red number, the numbers in red. And as you can see, as you actually add the high-C or OmniC scaffolding steps, you're actually reducing the number of broken scaffolds and total number of scaffold breaks as compared to the 10X-based assembly. Now, that is at a 10 kb resolution of the alignment. If you move to 300 kb, which is what we typically use for our chromosome evolution studies, you can see that the numbers are much, much lower in each of these categories, which tells us that most of the inconsistencies that are produced between these two assemblies involve small fragments in the alignment. Okay, and so one way in which we can evaluate these assemblies is to compare them. How do you know what is right? There is no perfect assembly to compare it to. So one of the things that we do is we use comparative information. Now, the closest relative to the narwhal is the beluga. It's the only other species in the family Monodontidae, and its divergence time from a narwhal is about 6 million years. And if we look at these different assemblies, we can see that there are actually very few differences across all of these assemblies at 300K resolution, but the fewest differences are produced by the most contiguous genomes, and particularly the genomes that are using OmniC and in longer, assembled with falcon-based assembly. And so you can look at that more carefully. So what we're interested in is why we're seeing some differences between OmniC and Hi-C. Can we evaluate the differences that we're seeing? And so Joanna, of course, went ahead and compared the falcon phase plus Hi-C to the curated assembly, and the OmniC to falcon phase and OmniC to the curated assembly. And you can see that there are some major structural differences between these assemblies. And I'll point you to two regions in the assembly that we're going to look at in a little bit more detail. An inversion that is located here at the very end of the chromosome in OmniC, and without using OmniC. And an inversion here, there's a region of alignment, but there's an inversion here that's in the falcon phase plus Hi-C. So that's one example. And here's another example of inversion that we're going to look at more carefully. And we do this in the same way that I showed you before, using the evolution highway tool. And this is really what we're calling, sort of in tongue-in-cheek, the directed evolution of genome assemblies. And so from left to right in this ideogram, you can see the 10x contigs overlaid onto the curated assembly. And here, these are just the contigs. So you see, literally hundreds, if not thousands, of individual contigs that align to this particular narwhal chromosome assembled using the 10x based reference. Then the scaffolded 10x, which is done with it's a supernova 2 assembly. You see much greater contiguity, and here's the evolution as you move out, adding each step of the way, reducing, in most cases, the number of scaffolds, increasing the contiguity, and of course the greatest improvements come when you add in the long-range scaffolding steps, either Hi-C or OmniC. And in this particular case, if the numbers are the same, this is if the numbers are the same, that represents a single scaffold. If the numbers are different, that means there's a break, and the break is right here. So this is 2 scaffolds in this particular subassembly. In the PacBio plus Hi-C assembly, this is all a single scaffold, and this is all a single scaffold in the PacBio plus OmniC. Only here you see the difference, the rearrangements. So this one is at the end of the chromosome, involves this inversion here, and this is the other inversion, which involves this color change here. And so how can you use this information to tell you which of these assemblies is correct, when in fact, all of the information that we have in looking at the subassemblies, because these involve different scaffolds, the subassemblies are in fact inconclusive. They can't help you to tell you which of these 2 is the correct version. However, we can use comparative information. And here are 3 different dolphin species, orca also in the Delphinae family, and the beluga. And as you can see, in this particular case, In this particular case, the comparative information supports the OmniC structure, so that it's an inversion relative to the 10x based assembly that is consistent in the PacBio OmniC based assemblies. So we would say in this case that it's more likely that this configuration is correct. Second case, very similar, not a lot of time left, so I need to move on through this and wrap this up, but a very similar case, but in this case, the high C structure was more supported using the comparative data. So conclusions here is no methodology is perfect, and we do not yet have a means of deciding which combination of methods produces the most accurate assembly overall. We know that scaffolding is giving us very high contiguity and a reasonably high level of agreement in the breaks and joints at lower resolution, but at higher resolution, there's more disagreement. We can use comparative information to compare the intermediate assemblies, allows us to pinpoint in many cases and resolve some of the inconsistencies in the assemblies and the problematic assembly regions, but verification of scaffold order and orientation in these flag regions it needs either to be looked at manually by looking at read coverage plots or high C contact maps, or confirmed using an independent method, for example, such as the optical maps. Now, I have to say that this is still a ongoing process. Directed evolution of proteins is really rather simple. Selection, amplification, significant, rather consecutive rounds, increasing our affinity of proteins, for example, for a particular target. But with genomes, our directed evolution looks more like this, where we're sort of constantly chasing our tail, trying to figure out which combination and which methodologies are the best. And we're currently looking at the Hi-Fi, as Mark mentioned at the beginning. Again, this is being supported back by PacBio and Dovetail, and a lot of information that we already have indicates that the assembly process can be greatly simplified by doing these Hi-Fi, very high accurate long reads on with PacBio and Sequel2 and combining with high C or base methods or OmniC. And this is a slide that was provided to me courtesy of Jonas Korlach, who I want to thank for this. And we are currently evaluating adding this to the group of assemblies for evaluation. And we have great hopes that this will greatly reduce the complexity and the cost. And I might add that the initial cost of the PacBio assembly of the assemblies, the VGP assemblies, was more than $50,000 for a 3 gig base genome, and we've got that now under $10,000, which is really quite remarkable, and will save us actually hundreds of millions of dollars, more than $200 million, in the first phase of the Earth BioGenome project. So why is all this important in the last 2 minutes? First, genome completeness is, completeness of the assembly is extremely important, and we just discovered that in our work looking at the ACE2 receptor and its relationship to its predicted binding ability for the SARS-CoV spike S protein. This was a study that was also put up on bioRxiv recently and is currently in peer review. And we know from looking at these assemblies that there were many errors in the gene models and in the genes that had to be corrected in order for us to do accurately and precisely at positive selection and accelerated regions or individual bases. So this is very important to us to have very highly accurate assemblies at the gene level. Of course, we are very interested in assemblies, in comparing assemblies at the chromosome level. And clearly having highly contiguous and accurate assemblies is absolutely essential for determining breakpoints, classifying those breakpoints, determining where on the evolutionary lineage between an ancestrally reconstructed genome and extant genomes those breakpoints occur. And so, again, high contiguity, high accuracy, chromosome scale assemblies is absolutely critical to this field of chromosome evolution research. And if you're involved in gene expression studies, also having these repeats assembled properly, all of the regulatory regions upstream, the intergenic regions and downstream are also important. In the same study by Marta Ferre that I just showed you previously in our chromosome evolution collaborative group, looking at how chromosome rearrangements affect gene expression is very much dependent on having high quality, contiguous, near error-free, if we can, genome assemblies. So I'm going to end there, and I'd like to thank again all of the this amazing team of people who have collaborated to produce these very detailed, painstaking work to evaluate different steps in the assembly process and the different technologies. Most of all, Joanna Damas, who I had the privilege of presenting her work today, the work of the Vertebrate Genomes Project, Genome K Consortium, Martin Nguyen, again, I mentioned him at Harvard Dental School, the curation team at Sanger, the assembly group at Dovetail, Jonas Korlak at PacBio, and our collaborators in the chromosome assembly collaborative, Dennis Larkin and Marta Ferre, who's now at the University of Kent. I'll stop there, and I still have 6 minutes, it looks like, for questions. And so please go ahead, Mark. Can you handle
great.
this? Okay.
Yeah, thank you so much, Harris. Fascinating. Yeah, we have several questions, and to the attendees that are still on the call, if you have a question, please type it into the Q&A tab. It should be at the bottom of your screen. So we'll go through these in order here. The first question, Harris, is, did you compare the combination of PacBio and BioNano with the combination of PacBio and Hi-C?
We haven't done that yet, but we're in the process of generating the CMAPs for the Narwhal assembly, so we will be able to do that as well. Okay,
great. Second question, is there any possibility of mosaicism contributing to apparent inconsistencies?
I suppose that is possible. I want to say also that when we looked at these assemblies, we have compared a phased assembly to a non-phased assembly. So the 10X assembly was not phased, and the Falcon, it was a phased assembly. So many of the differences could be due to phasing.
All right. Here's a question. This is a little bit lengthy, but I'll read it out. Thanks for the very interesting and complete presentation. I have two questions about annotation. Despite significant improvements, PacBio long reads still have a high error rate for indels, which can, even after polishing, heavily affect annotation due to the introduction of frame shifts. This can also make the affected loci to be undetected, for example, by Busco algorithms. What has been your experience with this problem, and how did you approach it? That's the first question, and then there's a second part.
Yeah, thanks very much for that question. And we have not analyzed the assemblies at that level, but I'd urge you to look at the VGP paper, the landmark paper that was put up on bioRxiv, which does address that question to some degree.
Okay, and then the second part of that is, in line with that, might some of the duplications you detect be actually fragments of a same loci split or non-contiguous due to indels and thus annotated as separate sequences?
Yeah, I think I understand the question. And these inconsistencies and fine scale differences can be, there are many different possible error sources. And as I've mentioned, there's no such thing as a perfect genome, but we strive toward a perfect genome. And I do believe that the new hi-fi technology, given the way that it works and the much lower error rate, is going to improve assemblies at that fine structure level and eliminate many of these inconsistencies, problems with small indels and the alignments that we're seeing.
Sex chromosomes are traditionally very challenging to assemble, and for you guys to have hopefully complete assemblies, do you have a strategy, or is there even a way to do it properly with the current technology?
I was hoping you wouldn't ask that question, Mark. But so I think for the X chromosome, we're doing quite well in the vertebrates, and that would include, you know, the Z chromosome in birds as well. You know, the Y chromosome is more problematic, and so of course having higher coverage and longer reads is a great advantage. But you know, right now we're not systematically looking. Our group is not systematically looking at the sex chromosomes. The standard that we're using for VGP and EBP is to have at least one of the sex chromosomes assembled to the quality metrics that we have described. But we have found that it is very difficult for many species at using those quality standards to achieve the smaller, the Y chromosome or the W chromosome, high degree of continuity.
Mm-hmm. Mm-hmm. Great, thank you. There is another question. I think we're at the top of the hour, so we'll take one more question here. Given limited DNA and budget, what approach would give the best BOOSCO scores per dollar?
Yeah, so I'm gonna limited DNA now is, yeah, it's really, you know, actually specifying the amount when you mean limited, because in the beginning, you know, that's one of the reasons we wanted to simplify the pipeline. You know, BioNano requires a lot of DNA, and the original PacBio protocols required a lot of DNA, but now, you know, PacBio, as we've seen, you know, can be done with nanogram quantities. You know, the head of an insect or the leg of an insect will produce an excellent assembly. And so for most multicellular animals, I think that it's no longer an issue to be producing long read assemblies. So I would always shoot for the long read assembly if you can. And then what you do after that in terms of scaffolding certainly might depend on how much DNA, you know, how much high quality, high molecular weight DNA that you have. You know, right now, if you have that DNA, I would use one of enough DNA. I would certainly use, you know, one of the Hi-C based protocols or OmniC to scaffold up the PacBio contigs. Now, that can be done. You know, the differences in cost of, say, using 10x or just a, you know, a short read assembly are you know, are really marginal these days. And we're not talking about from, you know, $10,000 to $50,000. You know, we might be talking from maybe $1,000 to $5,000 or something like that. And so I think it's worth all the effort, if you're sequencing a genome de novo, it hasn't been sequenced before, to put the extra resources into the long, you know, the higher quality assembly. You're going to be better off in the long run, because without it, as Eric Jarvis and Tu Lee and others have shown in the Vertebrate Genomes Project Consortium, you run into lots of problems with individual genes if you have fragmented assemblies.